you can present longer if you like. And we'll leave about 15 minutes at the end for the question and answer. For our virtual audience, throughout the presentation, please using the Q&A feature at the bottom of your screen, not the chat, and submit your questions. I hope you have been using Zoom fair enough already. We'll try to answer as many questions as possible during the Q&A sessions at the end for live audience, and then you can choose a chance asking questions during the Q&A as well. So today is really my honor to and a pleasure to welcome Professor Vicki Colvin. Professor Colvin is Victor Kriebel Professor of Chemistry and Engineering at Brown University and Director of their Bioengineering program. And really, in the past 3 or 4 years, I got to know Vicki, and we both are very passionate about doing something, change the diversity, equity, and the inclusion. So we're very, very pleased to also working with Vicki in NSF proposal, AGAR proposal, and keep our finger crossed. If we have that one, I'm really just looking forward, right? So that's not on the script. So now back to the script. Her research explores how complex materials in both composition and dimensions interact with the environment and living systems. This fundamental knowledge lays foundation for her group's development technologies, which using new materials in both medical and environmental applications. So examples including electromagnetically active particles in treating disease and using perpetual antioxidants for mediating foreign body response to implanted medical devices. She published over 200 peer-reviewed papers, holds 7 patents, and is a fellow of MB and also fellow of AAS. She has been named as Sloan Foundation Fellow, a chemistry of materials highly cited researcher, and currently serves as associate editor of the ACS journal Nano Letters. She received her undergraduate degree in chemistry and physics from Stanford University, and her PhD is in chemistry from UC Berkeley, and has been spent a lot of time in the East Coast for quite a while now, right? So I'm pretty sure you do not miss the snow in California. And let's give a welcome to Professor Colvin. Thank you.
Hear me? Is it on? Yes. All right. Well, it's a real pleasure to be here at Columbia. It's been a great day so far. I'm looking forward to more conversations this afternoon, and I want to thank Ed for having me come down. It's not that far. I'm from Providence, Rhode Island, where Brown is, if you didn't know, only a couple of hours by train. So it's a real honor and pleasure to speak with you all today. So what I'm going to try to do, and I'll try to keep it as short as I can, so you'll see me skip a lot of chemistry, is tell you a little bit about my research and some of my perspectives on how really amazing materials could change how we solve problems in biomedical engineering. So if I have a higher level pitch, it's that I hope that biomedical engineers everywhere, even if you're not a chemist, think hard about really exploiting some of the amazing materials that chemists know how to make these days and using them to solve some really tough problems. So let me see if I can get this to work. Does it work? Yes. All right, so I'm going to start by showing you a picture of your iPhone, or an older iPhone, or Samsung if you like. Many of you have them in your hands now. Put them away. If you look inside of this device, which I would argue has changed how we operate as human beings, what you're going to find, I have to figure out which button to use, is the following: a really complex set of systems that all work together to make this concept of communication possible. But even and I think there's some interesting analogies with biomedical engineering. We build complicated systems that have a lot of different components, and we need large teams of people with different expertise to make them work. But if you dig down another level and you ask yourself the question, what atoms are in that system? This is what you're going to find. By no means is this a system constructed from carbon, hydrogen, and oxygen. Instead, it has a lot of complicated device structures that permit it to be lightweight and efficient. It spans most of large fractions of the periodic table. And so if we needed all of the periodic table to solve this problem, which we all agree is a good one to solve, I can actually, some days I wonder if cell phones are a good thing, but I'm old enough to remember when they didn't exist. We probably are going to need the whole periodic table to solve the problems of biomedical engineering. And so what I'm showing you here is from our National Academy of Engineering in the United States. They've laid out some grand challenges, and many of the labs I've talked to here and I know are working hard in Columbia, things like how do you engineer a better medicine to cure disease? Or how do you use all of the information we have on patients, maybe add new information from wearables so that we can better diagnose and understand the progress of whatever disease or treatment they have? And how can you really diagnose things without having to cut somebody open and do so with a high level of confidence? These are grand challenges that we have as a society. And I think my thesis is that you're going to need to think broadly about materials to solve those problems. And you probably can't pull off-the-shelf polymers and off-the-shelf components and hope to really have impact in all of these areas. So one of the things that's important is this intersection between materials and biomedical engineering. And really, what are some of the latest physics understandings we have of complicated, let's say, oxides and how they could be applied to some of these problems? So that's what I like to think about. And I have a bunch of areas of my research. We make materials, we solve interesting problems. One thing I won't be talking about today is a fair bit of work in MRI. Some clinical, some preclinical activity focused on how you create contrast agents that give you more and detailed information, particularly for diagnosis of disease. We also have work I am going to talk about today in neuroengineering. I'm so happy to talk to a neuroengineer this morning. There's a few here. And I've been fortunate at Brown to be able to work in a collaborative environment with people who are really doing sort of the amazing work that neuroengineering is now making possible and understanding and manipulating our brain. And I'll talk about how materials play a role there. And then I'll turn to probably the last part of my talk. We'll focus on magnetic materials. Many of you know magnetic particles and what you can do with them. So I'll sort of bring you up to speed on what's going on in the last 5 or 10 years in this area. That's really sort of changing some of the opportunities for applications. So I want to start with a neuroengineering application. And this work is in collaboration with Dave Burton's group, Dr. Elena Atherton and Dr. Yue Hu. So as you know, in all these cases, collaboration is one of the best things to do because you get to learn new stuff. So one of the things I know is I'm a huge science fiction buff. Is anybody else going to raise their hand for me and say they like that? Thank you. Thank you. It makes me feel connected to the audience. I love sci-fi. This is a great, I don't know if anybody's watching this one. It's called Peripheral. I don't know, it's on net. It's great. You should watch it. It's based off a really good Mel Gibson novel. Anyhow, what you're looking at is one of the main characters who has implants in his spinal cord that help him manipulate and sense his environment in novel ways. And, you know, we can say ethically if this is a good or bad thing, but it's certainly really cool. And what you could do if you had this interface between machines and people is really astonishing, both for disease. And then if you want to write a cool sci-fi, you can as well. And, you know, companies are trying to do it. Of course, Neuralink, I'm sure many of you have heard of, are trying to do the same thing. They're trying to build devices that integrate with our nervous system and even our brain to manipulate it, maybe cure disease, but also many, many other possibilities. And so as looking at this prospect, it becomes interesting to say, okay, what are you going to actually have to do to make it possible? Because it's probably for certain diseases a really good thing. And it's already happening. So this isn't completely sci-fi. So over on the left, this is an example of an implant that I actually wear, which is a glucose sensor. And that implant has to only last 2 weeks. But it can sense glucose in my body. It's not a nervous system, but it does need electrical access to my body, you know, to the subcutaneous region. Cochlear impacts, another amazing example of a permanent implant. And finally, neural implants. This is a picture from BrainGate at Brown University, where they were one of the first teams to use neural implants to actually move, basically somebody who's paralyzed to move that implant around. And all of these cases are really cool because they require that you build a device that isn't just sitting in the body for mechanical purposes, but is actually fully integrated electrically and or chemically with the environment. And that actually really puts a high bar on an engineer who wants to build one of these devices, especially if you're doing it in people. Because usually you want, especially with the neural systems, you want them to be in there for decades. And one of the biggest problems is when you have to interface these devices with the body, of course, is that the body doesn't like it. It treats it like what it is, a foreign body. And that's going to block the application. It's the #1 limitation to neural recording devices in applications like paralysis or other diseases. There's some really cool examples now where these neural implants are helping people to speak who have locked-in diseases. But the problem is they just don't last very long. And why is that? Many of you will know the reason. And before I get there, this is an example of a neural electrode from Brown. So in that woman's brain was something called the Utah microelectrode I show here. This has got to be the scariest picture I've ever shown in a talk. It's these teeth, right? It just does not look like this is going to be very biocompatible. And answer, it's not. But it goes into the brain, allows you to do neural recording very effectively, and it is FDA approved for certain cases. The problem with this system is that the biology, of course, is really drastic. And while it can detect the electrical signals from hundreds of neurons, the problem is it kills the neurons in the near electrode region. So what I'm showing you here is kind of a picture. These don't go very far, just into the cortex. And once there, over time, you get neuronal death. And over on the right, I'll talk more about this model, is a cortical tissue where you can see it's sort of black near the tissue. You don't see any of the red stain, which corresponds to the neurons. And that's the neuronal death that happens not from the insertion process, but from the chronic implantation of this foreign body. So the brain itself protects itself, doesn't build a capsule like it might in other parts of the body, but it certainly reduces the electrical contact. And of course, the neuronal loss has other consequences. So if we're going to use these types of devices that really need to be connected, we have to figure out how to keep this biology from happening, at least not at the time scales that it is here. And so the concept, as many of you know, if you put something foreign in the human body, there is a series of cascading reactions based on immunology. This is one that's just from the conventional immune system, but the brain has similar analogies, where a whole cascade of events happens that ends usually in walling off, where you sort of protect your body from whatever it is you put inside of it. And the most important thing about the process from my perspective is that oxidative stress is one of the key early indicators of something being wrong. And so if you look at this graph, which is a function of time of many, many different inflammatory and oxidative markers, you'll see that ROS and a related nitrogen, reactive nitrogen, both peak. And those are often considered the key indicators for recruiting and developing the rest of this immune response. So one of the ways you can prolong lifetime implants that are recording is you just flood the patient with a systemic anti-inflammatory. And that's going to be really hard to do for years, something like prednisone. So you need a different way of attacking it, and ideally a local way, something that's only affected near the actual implant. And so the strategy that I'll talk about is reduction in oxidative stress, and to do so in a very intentional way with a material that is something called a perpetual antioxidant. So if you lower ROS, and this has been shown in animals, what you're able to do in these implants is they will live a lot longer, and you'll sort of short circuit these responses. So the material that we're going to use is an interesting material. It's not one you're going to know necessarily. It's something called cerium oxide. So cerium comes from the bottom of the periodic table. It's a lanthanide, and we can make nanoparticles of it. It's sort of the core capability of my lab. The important thing about the ceria, though, is that it's got a really interesting property, and that the oxygen in its crystalline lattice is very back and forth with the solution. It's actually a sponge for oxygen. It's a material that you would find in your catalytic converter, for example. And it's used a lot at high temperatures for sort of its oxygen buffering capacities. And in the last decade, or probably 2 decades at this point, it's been applied in some very interesting biomedical applications, because what it can do, up there are some EM pictures, I won't go into the crystal structure, but I will stop here, is that when it's in the presence of reactive oxygen species, it actually will oxidize. It's very aggressive at pulling those oxygen atoms out of a biological environment. And it's a cerium 3 to cerium 4 conversion. But over time, it reacts with donors in the biological environment. It goes back to its stable cerium-3 form, which is what it likes to be when it's in sort of ambient conditions in the body. So it's reactive enough, but then it holds onto it, buffers it, and then eventually recycles back. The recycling time can take up to about 1 to 3 weeks, and then it goes back to being reactive. So it's sort of like a holding pen for reactive oxygen species until it can do a second set of reactions that are less damaging and return to its starting state. So the term we use for this is perpetual antioxidant. And if you think about the challenge of delivering an antioxidant to, let's say, one of those electrodes for decades, is you're going to just need grams of an antioxidant if you're going to use it, right? How are you going to put that on board an electrode? So if you go to inorganic materials like these Ceria systems, they can sit there and they can basically be a cycle that allows those species to just overall drop. And so it's really, really promising for chronic applications of this type. All right. I'm doing this. I will do this. Hopefully, it has now stopped. Any suggestions? I think you came and somebody changed it. There we go. Yay. All right, I'm going to skip this slide, except to say there's some really amazing chemistry that my student Yue Hu did to actually, what's interesting about this material, these are freeze-dried samples of the cerium oxides. They go right into water. They are stabilized by very carefully designed polymers that activate this perpetual antioxidant behavior. So it's an interesting trick. And if you're a materials person, we can talk offline. How do you put a polymer coating on an inorganic system and still allow access to the surface? That is a problem we can solve, but it does require a lot of thoughts about what those materials might be. Okay, let's see, good. So one of the ways that we measure the success of these systems is we actually just treat them and study their antioxidant recycling capacities. And so some of these materials are very, very good at it, and they don't last forever, but they certainly can absorb huge quantities of reactive oxygen species multiple times. And so they go back and forth, and they can be challenged over the span of days or even months. All right, so how are we going to tie that to these Utah microelectrodes? So the access for me are anything that's associated with a recording system that's not the actual tip of the electrode. So there's a lot of polymer coatings that exist in anything you're going to put in the brain. And that's actually the site that we focus on for introduction of these perpetual antioxidants. So we form these systems in a slow release polymer. We can tune those release characteristics through a variety of means, and we can measure the ceria coming off in the media that we're using. We've also thought about attacking them down and tethering them, which we are exploring now for really, really chronic use. But right now we were just trying to say, hey, in some of the models we're working with, how well do our ceria perform, and does it correlate with their ex vivo activities? So we have a great model, and this is really the work of Elena Atherton, who worked between Dave Borton's group and my own, because one of the challenges is if you're trying to mediate the chronic implantation problems, how are you going to study it? I mean, we can make hundreds of different types of these nanoparticles. We want to know which one's the best. And that's not an easy thing if what you require is, months of a primate experiment. So what we really had to do that was actually very useful is develop a sort of basically a cortical tissue model for looking at implantation. So in this model, you basically take cortical cells and you differentiate and grow up a microtissue. What's very interesting that Elena discovered, and I'm going to use my cursor, is that they have to be under some mechanical stress. So what you're seeing here, this picture, is a 3D fabricated holder that allows these tissues to kind of grow and develop in the way one would want. That was a really important insight. What you see here is the wire, where the wire will be placed. So after you collect these, you grow the microtissue, you place the wire, and you can do a lot of different biomarker detections. You can do live cell imaging. You can also look at endpoints of oxidative stress, a variety of types. And so we can show down here, you can see an H and I, both the stain on, in this case, neurons, as well as calcium activity. So we're able to damage these microtissues and sort of ask the question, what happens? And the good news is, as we expected, we measure lipid peroxidation, for example. When you insert a micro wire into these things, you get a lot of oxidative stress, as you would expect. It's a foreign body. The good news is about 400 microns away from the surface of the implant, it tends to die down, which is consistent with others who have studied neuronal death near these implants. We can also measure neuronal volume. So we looked at over 8 different biomarkers. And again, if you put a wire in this tissue or probably in a brain, you have a lot of neuronal death due to oxidative stress. So when we put the Cerium nanoparticles, and this is our best material, the one that had been optimized for its perpetual capabilities, we're able to completely mitigate this oxidative stress. And all of those biomarkers go back to being normal. So it worked as we might expect. It's a highly, really good antioxidant. We looked at things like neuronal density near the implant and found that with Cerium, we didn't see. So you can see in this picture, a lot of death of neurons, which are the red stain. And if we have Cerium present, we're able to protect against that because we're mitigating that ROS response. I'm not going to go through all the biomarkers, but I will focus on this one because this taught me a lot. So this is actually data from the calcium imaging. So these cortical tissues actually show firing, and you can analyze the sort of node-to-node interactions among the neurons that are present. And so in a healthy set of neurons, what's really interesting is you don't get every neuron connecting to another neuron. And I'm sure I have people in here who know this better than me, but you find sort of groupings of neurons that fire together. And that's actually considered a sign of healthy neuronal behavior. It's sort of a subacute toxicity threshold. Once you put that wire in, it all goes away. They still fire, but their organization is completely lost. And this will happen before you see a lot of loss of neuronal volume. So it's just the presence of the wire just sort of skews everything. However, if you've got the protective antioxidant system in, you're able to recoup and show that your neuronal nodal patterns remain the same. So that's an interesting finding that actually had some consequences for people who study these properties. Often these electrodes are used to study node-node interactions, characterize what this is. So the fact that the wire could affect those properties alone was interesting, but certainly with the right material, you can mitigate against that. So these are kind of interesting systems. This is my end of that story. And I'll just conclude by saying we're basically developing, we have this great model, those cortical microtissues can be done at scale, high throughput, sort of. So we can evaluate many different combinations of materials. And we're looking at co-delivery of other agents. So we're probably going to be putting in some biomolecular targets as well as the nanoparticles. And we're also looking at how far out the nanoparticles go. We have about a 400 micron area to protect. So the question is, can we match perfectly the diffusion of our materials over that scale? So there's a lot to do there, but I wanted to show you an example of this exotic cerium oxide and what you can do with it. I want to move next to magnetic systems. And this is kind of the bulk of what I wanted to talk about today. Again, I'm back to sci-fi. Has anybody seen this? No? Anybody's? No. I have a okay. Some sci-fi, maybe not. So this is from basically a Star Trek, and this is the Borg, and they have a tractor beam that is pulling this Enterprise towards them. And so this concept of movement through space without contact is a powerful one, right? And this always has captured my imagination. Magnetism is one of the most difficult topics that I've ever studied, which means I've spent my life trying to understand it better. So this project goes right hand in hand with it. So what I want to do is talk about the role of magnetic materials in biomedical engineering and give you some examples. I'm going to first talk about the material, and then we're going to talk a little bit about the applications. 3 things you can do. If you have magnets inside of your body and they're small enough, you can move things around. So this concept of, for example, if you've done a systemic delivery and you want to concentrate a material at a very particular area, maybe you could do it magnetically. If the delivery agent was magnetic, that's moving a magnetic thing in the direction of a field gradient. Magnetic hypothermia, the concept that if you oscillate the magnetic field, you can heat up the material. And finally, imaging, the fact that any magnetic material is going to interact in ways with magnetic fields that allow it to be imaged, both magnetic resonance imaging as well as magnetic particle imaging. So all of these things are possible with magnetic particles because particles are small enough that you can integrate them with human body tissue and ideally target and place them where you want to place them. So it's a very compelling concept. And interestingly enough, I would argue that magnetism through the development of MRI was the first case study of sort of physics really integrating with medicine in a deep way. And the fact that there are huge magnetic fields and MRI systems everywhere in hospitals, one of the consequences is that there's an interest, or at least not a resistance, to using magnetic fields in other ways. So I think this connection and the power of using a magnet to at least image has been demonstrated. So this particular direction is one that I think when I talk to clinicians, they don't laugh me out of their office sometimes. Sometimes they do, but all right. So our problem with the application is very fundamental. While there's a lot of cool things you can do with them, I can't point you to any commercial example of a successful magnetic material that's been clinically translated. I can't. Even in MRI, where there is one that's been FDA approved. And the fundamental problem is, well, in that case, it's another story, but the problem is that our materials are magnetic, but they're really crummy magnets. So when you make a magnetic material, especially a small one, that you need, it needs to be small to get into the body and interact with cells, so ideally smaller than a cell, it's not going to be a great magnet, which means if you want to do any of those cool things with it, you're going to have to apply a magnetic field that's many Teslas. Think the size of an MRI system. So you've got this huge need for large magnetic fields, and the fundamental parameter is susceptibility. It's how much does the system magnetize as a function of applied field. And the susceptibilities of our systems are thought to be fixed by their composition and crystal structure. So they're actually really tiny, and you need ginormous magnetic fields. So what my group has been interested in doing is actually changing the paradigm where we focus not on large field applications, but really, really, really small scale. So kind of our challenge is to make a magnetic material, it's likely a particle, that will be fully magnetized, but to do so at 10 to 100 times the Earth's magnetic field, to really drop the sensitivity of the material to the magnet. So to understand that, you're gonna have to learn a little bit about why things are not easily magnetized. And the consequence of that is here over the left, I'll point to this. This is a system in Germany, which was done in the clinical trials for magnetic hypothermia. And this is the size of the magnetic system they needed to apply it. And the cost was on par with an MRI. So it's a very expensive treatment. And it's really because the materials they were using are just not very sensitive. So what we want is something that's going to have susceptibilities that are at least 2 orders of magnitude better. And in doing so, we think we would open up our ability to use a magnetic field in some of the ways I talked about. So a little bit about why they're crummy magnets. So magnetic particles are going to be at 0 field, not magnetic. But as you apply a field, all of their spins are going to align, if you remember back to physics. And that alignment and the size is going to be proportional to their volume. And so the saturation magnetization at high fields is everything's magnetized, and that's great. The problem is, why don't they magnetize fully at very, very small fields? What's the barrier to those spins rotating, in this case, anti-parallel to the applied field? And the reason is really fundamental. It's that our systems have a crystal lattice, and that crystal lattice creates a barrier to the rotation of the spins. And as a result, their susceptibilities are very, very large. So it's hard, or sorry, very small. It's hard to make them magnetized because those spins are working against basically the crystallographic lattice itself. So it's thought to be embedded in the system that crystal anisotropy is going to limit how sensitive magnetic materials are going to be, which means any crystal system has got its intrinsic anisotropy, and you can't beat that. But you can. And the way you're going to do it is to imagine a porous material. So this is a large single domain magnet with a bunch of spins pointing in one direction. And if you put pores into this crystal, so you basically make gaps, the spins in the different parts of this porous system are somewhat aligned, which you want, but the pores themselves lower those anisotropy, those barriers to rotation. And there's one of the ways to make highly susceptible porous bulk magnets is to actually introduce pores. So our question is, could we do this on the nanoscale? And the hypothesis was that we could decrease some of the interactions that were causing these barriers to rotation by producing a system that's highly crystalline. We can't give that up because that has to do with the ultimate magnetization. But could we put enough defects or pores into the system to allow the spin rotations to occur more easily? So here's what we kind of needed to realize this vision. We definitely wanted iron oxide because we are, biomedical engineers. So we'd like a system that's already been FDA approved, although hopefully some of the other systems that are coming up will make it too. So we're working with iron oxide, which is a really good magnetic material to work with. It's got a high saturation magnetization. We know it well. But our approach to making it, yes. Okay. Our approach to making it porous is to cluster. So each of these yellow balls is a single crystal, but those single crystals are oriented and attached during their formation process. And in doing so, they're very oriented across. So it's like they're all pointed the same way crystallographically, but they have holes where they're not perfectly cemented together. If you're able to achieve that, you can get very, very high susceptibilities. So I'll tell you how we do that. This is going to go really fast because it's a lot of chemistry. So let me try to get to the final end goal. I'll stop with this picture because I think it's the one you need to see. We produce these materials using a pretty conventional synthesis, actually not as good as some of our other syntheses of nanoparticles because we intentionally aggregate the particles together. So what you're looking at here is a cluster of nanoparticles. They have an overall dimension, but they also have a small primary dimension. And they are 50% porous. So there's a lot of internal surface area because they're not perfectly condensed. When you do that, you can control their size, which I'm not going to talk a lot about. But you can do studies of the crystallography of these packed aggregates. And one of the remarkable things about them is that they are oriented in every way. So even though this looks like a loose collection of randomly oriented things, it isn't. That orientation process that occurs in this chemistry when they're forming forces the crystal axes to point the same direction. And that oriented attachment, it's actually been known for decades to occur in other oxides, is crucial for this, for what I'm about to show you. So it means that we're able to cluster our particles in a very well-defined way. We don't want big aggregates, but we're able to do so with these holes. I'll get to my applications. And the good news is it's what we would expect. So when we make these clusters that have the holes, they're porous single crystals, their susceptibilities are substantially orders of magnitude higher than if you had a single crystal with no holes, and if you had smaller, just a single particle. I'll show you that data now. So this is a comparison of clusters and particles. These are field strengths that I'm showing here, which really are on the order of like 10 times the Earth's magnetic field. So very, very low field strengths. And overall, what we're able to do on the right is actually the data, and on the left is kind of a different view of the data. One is a ferrite and one is an iron oxide. We'll talk about this one. Is we're able to get very, very high susceptibilities, and this is in a solid phase. In a liquid phase, they can be an order of magnitude higher. So that really high susceptibility, but for very small cluster sizes, is perfect for the biomedical engineering applications. This is just another graph showing if you plot versus volume, you can actually see that there's a strong volume dependence to susceptibility, which we would predict. And that if we make them too big, they collapse to being permanently magnetized. And that's actually not what you want for any application in the body. So you really want something that's not magnetized at zero field. We have some excellent collaborators who've simulated this, which has been really important for our understanding. Like the size dependence is fascinating, but I want to talk about how we use them. So let me close with that. So the first thing you can do with these is if you've ever thought that magnetic particles will aggregate, well, they do. I just showed you some that do, but you can totally coat them so they don't aggregate. So over on the right, these are what the clusters look like in solution. They can be freeze dried, but then you can dissolve them up in whatever you want. And they have a polymer coating that has taken several student lifetimes in different ways to optimize. But what you're looking at are solutions that are in brine. So some of these solutions are, seawater. And what that means is colloidal stability, the fact that these solutions, especially the one on the far right here, still look clear and brown, mean the particles are not aggregating together. And even in the presence of a field, they won't. And so the particles are little aggregates, but in solution, they're like non-interacting particles. We achieve that by putting pretty high charge densities on the particle. We use a polysulfonate system that is actually approved for use in humans or close to it. So we think that's not a problem, but we're able to manipulate stability of the particles. And you'll notice the middle one, we've done this for 4 years. The middle one is not stable. You see it crashing out of solution. Anybody who's used nanoparticles knows that's like you put it in your media and they're done. You can't inject that or use that in any way. So one of our big goals is to make our materials very colloidally stable. And so we're able to achieve that even though they're very, very good magnets. Once we have those systems, we can do a lot of stuff. So now we're going to go through a couple of examples and hopefully maybe some of them would relate to things you're interested in. So one thing we can do, everybody wants to know about toxicology and what happens to nanoparticles. So this is one example where you can just do a 2D cell culture experiment and put in our materials and ask what does a cell do with them? And the answer is it depends a lot on the coating. If it's a polyethylene glycol coating, the cells won't do a darn thing with them. If it isn't, cells are going to take them up and they're going to put them in the lysosome. And so that's a very standard entry pattern. And for our applications, that's totally fine. And you can also conjugate. You can do a lot of toxicology in different ways on these systems. And the take home is that the particles get taken up in the cells, and you can also track them. So we can identify them with different dyes, which is very useful for a lot of live cell imaging applications. One of the first things that we did with them, this is work by Linlin Zhang at Rice University, who works with Gang Bao, who's one of my primary collaborators, is they used it. They're very interested in separating mesenchymal stem cells from media. And so what they were able to do in this experiment is show that you could tag and target these materials to MSCs. And what you're looking at is one of the letters of a toy. If anybody's ever put magnet letters on their whiteboards or on their refrigerators, they're very, very poor magnets. So we put the letter A underneath, and we were able to collect very efficiently the clusters in a matter of seconds. Where you just can't do that with a solid particle or with a particle that's a little smaller. So these porous materials have that going for them. They're very, very easy to capture. The forces that act on these materials scale with the susceptibility. So those peaked susceptibilities mean you can pull them out of solution easier. There is a volume dependence here, so you give up a little bit there, but overall the susceptibility wins. So we're able with extremely small field strengths to manipulate them, at least in these examples. Another really interesting case is whether we can use them for magnetic particle imaging. So I don't know if you've heard of MPI, but it's a background-free technique for imaging that's kind of emerging that uses basically application of magnetic field. And if you have something that's magnetized in the middle of it, it creates sort of a dimple in the applied field, which can be detected. So unlike MRI, which works in a very different way, and NPI is really detecting the actual iron oxide or magnetic particle in the system. And one of the challenges for NPI, and this is just some of the basic information about it, is that you don't really have very good agents that really give a big enough change in magnetization for the field strengths that they're operating at. So they also depend on susceptibility. So this is some work with Steve Corral at Berkeley, who does a lot of MPI, and he has a company that's set up to sell the MPI system. So that's the disclosure there. They have something called VivoTrax, which is their imaging agent for these systems. And we had predicted that we should have a substantially higher scattering function, which translates into resolution. That's how they end up using it. So indeed, when we gave him a lot of our samples and worked with him, we got very, very highly sensitive MPI detection. And ultimately, we were able to use that. Yes, we have one in vivo data set here. It's kind of disappointing if you want to see a disease. I'll show you one more in a second. But in this case, what we were able to do is show that we could get very high spatial resolution after clearance to the liver, which is what happens to these particles upon injection. These are not specifically targeted at this time. So it turns out I sort of asked the company, is this enough? Like you get a gain in resolution, and they're like, absolutely, you know, when can we sell them? Everybody wants to have that extra resolution or the enhanced imaging capabilities. So this will probably be something we'll be looking at trying to put in the hands of researchers. And if you don't know anything about MPI, you should look at it. It's got a lot of interesting applications to consider. Another application that I can't miss talking about is hypothermia. So one of the things I mentioned at the beginning is, of course, the concept that a magnetic nanoparticle, when subjected to an oscillating field, will heat up. And that's actually a really cool idea, and it's been used in some phenomenal ways. If you can put nanoparticles all in a tumor, you can heat it up and ablate it, for example. Same idea as photothermal ablation. The figure of merit for this system would be something called the specific absorption rate, which is how well do you absorb that oscillating field and translate it into a delta T. And as we expected, what I'm showing you here is as a function of cluster size, you see a peak in that effectiveness that really peaks right where our susceptibility does. So highly susceptible particles that are highly sensitive heat very efficiently. And it pretty much maps directly onto our measures of susceptibility over a range of different sizes. So one of the easiest things we did that a lot of people do is we just ran 2D cell culture, and we found, yes, indeed, we're able to kill cells when we heat them up at incredibly low fields. I don't know how many of you do cell culture here. Anybody? Any student do cell culture? You have an incubator, right? So it's hard to imagine how you would get a giant magnet inside of an incubator. And that's actually limited a lot of studies of magnetothermal heating. And so we're able to do that because we have these little battery powered devices, which are not too different from a magnetic stir plate. In any case, this would be your option before. You would have to use a very large coil and a lot of power in order to achieve heating. One of my students, I'm so proud of him, he's such an engineer, he built his own portable induction heater that we could use in small scale environments, like inside of a chamber, and he made it with a battery, so we didn't have to run a cable out of the system. So the point is that the ability to go to these lower fields means that we have the capacity to use much, much lighter weight and maybe even wearable systems to generate fields that can manipulate materials. And that was how we did a lot of our 2D cell culture. Of course, I'm going to skip this one. I want to really give a lot of credit to Gang Bao, but I will talk about the work that we've been doing with him in hypothermia and cancer treatment. And we just recently had a paper come out on this, but I'll tell you some of the background. So he, of course, looks at ways of doing in vivo treatment of tumors. And we do an intratumoral injection of these materials, which is actually a standard in the community that studies them. And in this cancer model, then you put them into their magnetothermal heater shown here, which is a large one, doesn't have to be that big. It's just what we have. And you see an enormous increase in temperature for these materials. And I think what's kind of nice about this is you might have seen some of this work before, because a number of groups have tried to do various aspects of this. So this is a pretty typical cancer model, rodent cancer model. And what's important to notice is that after 7 days, if you have both these really sensitive materials and you have an applied magnetic field, you can actually completely eradicate the tumor. And there's this, the follow-up histology supports it. And what's interesting about this is that this is one of the first cases where the magnetic field strengths were low enough that we are potentially capable of treating humans. You may not know this, but one of the issues that came up when they tried to move to human trials of magnetothermal heating is that there's actually a safety limit to AC magnetic fields, meaning we feel pain. They took a bunch of people in the 1950s and they put them in different frequencies of field and different field strengths. And there's this curve about you basically, if you have a higher frequency, you're more sensitive, you can just feel pain. And so a lot of the work that's done with hypothermia wouldn't translate to people because there would be a lot of pain response for those fields. So these fields are well below what we would predict the clinical threshold would be for these frequencies and these intensities. So it's an option that we might be able to get by what some of the early clinical trials in Europe found for the application of these systems. And we didn't see any evidence that there was a problem with the mice. And of course, there's a lot of toxicology and other work that would need to happen. Really interesting question is how to target these, which I'm happy to answer. And this is what we just talked about. So this is some of the existing data on magnetothermal systems. And so our system is definitely occurring at frequencies and field strengths that are putting it in the safe regime and are able to completely eradicate those tumors. I'll conclude with this one because I started with Neuro Engineering. So there's been a lot of work in using, we talked about electrical contact with the brain. What about magnetic? So in this particular example, the idea, which really is, Polina Neva and others are really driving this field of magnetogenetics or magnetic manipulation of the brain. So my role here is pretty small, but this is just such a cool project. I had to talk about it. And this is with Jacob Robinson at Rice University. So what we were able to do is generate these highly sensitive materials, and we tuned their frequency dependence. I won't tell you how, but we had one that heated up when frequencies were really high and one that heated up when frequencies were lower. And so we had two types of magnetic particles. And what you do with those is we can target those to very specific ion channels in the brain of developing fruit flies. So these fruit flies were treated with our materials. It's amazing what it's like to get a needle into there, but you can do it. And then they attach to the ion channels. And then when you apply the field, they either open the ion channels or not. And so we were able to show the two different particles. One of them is going to be heated at a different frequency. So with the same field strengths, but different frequency dependencies. Now, the other thing to keep in mind is our stuff is so sensitive that you can actually put the fruit flies on top of a coil. And the fringe field is large enough that you can access and affect those ion channels. So what Jacob's lab did after this treatment is they put the fruit flies on top of a coil. And they actually showed that if you played one frequency, which we put to one specific ion channel in the brain, the fruit fly wings opened and closed because that was the early area that we injected. In another one, we injected into more of a motor area that controlled how the fruit flies walk around. And so then they could walk around in a circle. So basically you had fruit flies, and depending on the frequency of the magnet, they either opened and closed their wings, or they walked around in a circle, or they did them back and forth and back and forth. So there's a great Nature Materials paper out on that if you want to take a look at it. I was going to show a video, but it would take too much time. But it's a really cool thing. And the point is, I don't know how you feel about magnetic fields manipulating behavior. It was pretty invasive to get the particles in there. But I think it's an interesting application of particles and fields. I don't know if you know, but transcranial magnetic stimulation is actually clinically used. So it sort of is possible that you could see an avenue for use in humans. It would not be crazy, but again, you've got this much distance between the applied field and maybe a region you want to go to in a human brain. So those materials need to be exquisitely sensitive to the applied field in order to have the biological effect you want, which is why we spent all this time on those porous particles. All right, so just to conclude, cerium nanoparticles are these really cool perpetual antioxidants that might be able to be integrated into electrically active devices to prolong their lifetime. And we also talked a little bit about porous nanoparticles. They are really, really good magnets if you put holes in single crystals. It's a little counterintuitive, but it's true. And because of that, you can ablate tumors at clinically safe fields. And you can also access neuronal stimulation using magnetic field configurations that are far different from those applied in the past. So with that, I'd like to acknowledge some amazing collaborators and thank all of you for your time. I'd be happy to take any questions.
We can unshare this. So, what is up here? All right, that's really a science, and now I understand why you like science fiction, because that's you're doing a real science fiction actually, it's actually working. So thank you so much for really fantastic. So before we get in a question and answer, I just want to call everybody's attention to our next hybrid seminar next week, March 10th with Matthew Thresh from Northwestern University. So please follow us on social media to receive the latest update. You can see our social media information on the slides. And you can always access registration information on our BME seminar of our BME seminar on the website, bme.columbia.edu. So okay, we have a few questions to get through, and I'm pretty sure our online, let's go with the live audience first, and then we answer a couple questions for the online. Yes.
So again, probably a fundamental question, but I don't understand why the spins are I guess create something that's more magnetic than ever do with the five or add more folds. It does seem very counterintuitive.
It does. Yeah, I think I'll give you an explanation, and it won't be perfect.
We need to repeat the question.
So let me repeat. It's a very fun, thank you for asking. I realized my audience, so I sort of skipped all of that. So let me go back. The question was, how is it putting holes in a single crystal makes it a better magnet? Fundamentally. So it has to be done in the right way. If you just add more holes, eventually it won't work. So the idea is when the spins move, right, you need them to collectively all point the same way. Got that? So if they're pointing this way, you put the field the other way, you want them to rotate, right? Okay. So the price you pay for the rotation depends on how organized they are going from pointing here to pointing here. When I put holes in, what the theorists found is they watched the trajectories of the spins. What it did with holes is one little part of the lattice moved, and then another part, and then another part, and another part. So they kind of moved through that transition in a separate way, kind of more like the smaller particles, and that really lowered the barrier. But then they all ended up pointing the same way. Okay, but if you put too many holes in, then you don't have enough spins to make a magnet, right? So it's this interesting, does that help? It's this transition of how do the spins, so they're not coherently rotating through the transition. Great.
So we have a few questions online.
Sure.
I'm going to jump ahead because I have a question too.
Sure.
So in the beginning, you talked about you mentioned anyway, cochlear implants. And I know for cochlear, I'm just wondering if they've used this stevia for that application at all.
Great question. I guess I don't have to repeat it. So the question, if you didn't hear it, is how has the Cerium been used for cochlear implants? And so what's interesting is cochlear implants do have a lifetime. They only last, I guess, up to a decade before you have to replace them. And they typically can prolong the lifetime if a patient will take something like prednisone or anti-inflammatory systemic. So there has not been any focus on local delivery for cochlear, but there should be. And Cerium could be an interesting option. There are other sort of biomolecule strategies you could look at too. It's up against the same issue as people want it to last for a long time. So how do you put on board all of it? But right now, to my knowledge, there isn't any application for that, but there could be.
Yeah, I mean, I know they use dexamethasone.
Right.
But then you can't, I think it's just for like the initial implantation.
Right.
And, but yeah, anyway, very, very cool.
Yeah, so maybe you could load up dexamethasone. I did a calculation of how much you would need once, and it was several grams. So I was like, it depends on how long, right? And what the protection radius is. But it's certainly a possibility. I'm surprised there hasn't been more engineering of these near device areas for things like mitigation of FBRs or foreign body response.
Yeah.
So I think it's an opportunity for biomedical engineering broadly.
Right. So now I guess I will, unless we have another question. Oh, yeah.
Yeah.
Great talk. I just wanted to ask you about the animal models where you've injected the magnetic beads. Do you usually keep the magnetic beads in the animals, or do you extract them afterwards?
No, we keep them. So I haven't shown data here. They're an intratumoral injection, which we're very interested in local delivery, as you can see both of these examples. So they're intratumorally injected and they're left behind, but we have done small animal MRI to figure out where they go. They are dark in MRI, and roughly 70% stay retained in the tumor environment, and the rest are cleared through the RAS in the liver. So they do get cleared. But we haven't done long-term biodistribution away from the tumor. And it's not clear to me that that's, the animal models are maybe not, I would want a different animal model to address perhaps more clinically relevant questions. This is just a sort of quick check, but it's an excellent question. Yes.
oxide, and we saw early acceptance for neural models in vitro.
Mhm.
But are there any concerns of when you have to transition in vivo as that environment is more diverse and less controlled?
Yeah, always a question. In vitro tissue, cortical tissue models, and then in animal, right? There's a ton of concerns because we all know that there are limitations to in vitro. I think that from my perspective, the question is not so much the biology and chemistry of the near environment. I think that won't be our issue. I think the issue is the long-term fate of the ciria. So if the ciria is diffusing throughout the brain and doesn't get taken up by microglia, which is what our early data indicates that it will, over time eventually end up in that cell type, then there are questions about the impact of, in principle, it would be no big deal, an antioxidant floating around the cell. But once they get taken up by a cell, they're going to be subjected to an acidic environment. They should be fine. We have no evidence that there's any cerium release or issue, but that is, neurons last forever. You don't really know. So I think the long-term application, one of the things we've been talking about in this project is actually tethering the ciria, if it makes any sense, basically tacking them down with one polymer or couple. So they go out, but they're like tethered to the electrode area. So we've been discussing things we can do to mitigate their long-term transport, just because we think that would be more acceptable safety-wise. But yeah, it's a huge set of questions, what would happen in an animal. But I don't see any obvious reason other than the long-term safety questions. And I can't try out 80 different formulations in an animal. So now I know the formulation I'd want to try at least. Great question. Thank you.
So I think we have a bunch of questions here, but I think the best question was basically that question.
Ah.
And the others, I think you could probably answer offline, like if you share the fruit fly paper. and
Oh,
stuff like
yeah.
that. So I think it's good to go ahead to another student.
Sure.
Yeah, I was just gonna ask, given that kind of the release of reactive oxygen species is a known cellular mechanism for killing intracellular bacteria, is it possible that the nanoparticles that are taking up reactive oxygen species would then leave those cells susceptible to bacterial infection?
Yeah, that's a really good question. I guess, would you be worried about that in the brain as much?
Because it depends on whether the bacteria.
Yeah, I think that's a really good point. So I think if I were to restate your concern, which is, I feel like I'm in an NIH panel, so great job. It's that ROS is the body's way of signaling its immune response, right? So if you knock out the ROS species, you're going to knock out this immune signaling process. I think my answer would be yes, if there was an area for infection or if a local area was getting treated, it would be reduced. But I will flip it and say, this is why you have to do local delivery. It's crazy that they're treating people with large quantities of antioxidants or anti-inflammatory systemically, right? I mean, I agree, locally, it's not a good idea, but if we can contain it to that region where it's needed, I think that might be the mitigation response. There's a disease, so you wouldn't do it sort of just for kicks. But it's an excellent, I hadn't thought about the specific question of the bacterial infection. I think for something like the glucose sensor, which we're also working on, that's a case where, you're doing the injection, you could easily introduce bacteria. If you basically neutralize the early stage immune response, yeah, that would be a problem. Absolutely. Although I didn't talk about it, just since you asked about immunology, one of the findings that recently Gang Bao and our groups published was looking at in detail the immunology of those tumor cells. So when you put a nanoparticle in these tumors, you activate the immune response in ways that are very good. So the nanoparticles for the iron oxide activated, and that was a good thing. So it would be interesting for ciria outside of the brain, what if you would see a similar activation process. It's thought to correlate just with a bunch of nanoparticles being there. It's not about their composition necessarily. But it's an interesting thing to think about.
So I think, any last question? Okay,
Oh,
I
wait.
saw your hand before.
Yes.
How, when you mentioned that a lot of graduate students build a self-side magnetic,
Mhm.
how customized
Really customizable, meaning like, so we are looking at, so the fact that we can do it at lower magnetic field, so they could be different sizes, you can have coils, you can have permanent magnets. You can rotate the field really slowly like a stir bar thing. In some cases, people want that. So they're pretty customizable. If you had, I think what gets hard is if you're dealing with a very large animal and you want a field that's like this far away. That's the hard, like everything we can do in small animals and cell culture is easy. And there's quite a bit of customization. There's a really good paper by Polina I don't know how to say it, Akivov from MIT, where she went through kind of the different configurations of how to construct magnetic field devices. And we can do most of those plus a few others just because we're so sensitive. So we can do quite a bit pretty easily. The student who built that did it. I couldn't believe it. We had trouble with our stockroom. He just bought everything off of Amazon for like $5. And my stockroom was like, wait a second, what is this? So it was relatively easy for that scale of activity. We're happy to share that design with you.
So I think we've come to the overtime even by a couple of minutes, so let's thank.
Thank you. Oh, okay.